Thanks, Sue. So first of all, thanks for giving me the chance to present this talk here. It was actually interesting to think about the topic and to try to wrap up some data. I also discussed with some people like Jan Molenaar, Johannes Schulte, and Angelica Eggert. So thanks to them for sharing some thoughts how I would design this presentation, because, well, overall we know there are not so many recurrent mutations in neuroblastoma, but at the same time I think we have enormous opportunities in the near future with the information that is coming forward. And definitely also at this meeting there's a lot of new biology and a lot of ideas about possible novel venues for targeting. So that's important to bring that all together in this talk. During the talk of Neil Rosen, it was already indicated that, of course, with the novel compounds, just as we did with the chemotherapy, it doesn't make sense to use single compound approaches. And I just took 2 quotes from a recent review here that single agent targeted therapies don't make that much sense, and that we have to think further. So I think we have to try to exploit the flurry of further cancer omics that is coming towards us. At the same time, and I think that was also really nicely addressed during in the second part of the talk of Neil Rosen, that we really have to try to understand not only the signaling pathways, but also how tumors are escaping the effects of drugging and how resistance mechanisms occur. And I will touch very, very briefly, as this is not my area of expertise, but some food for thought on future drug designs and novel therapies. And I understood there was a parallel workshop while we were doing the epigenetic workshop on Glenn Marshall on how to design therapies and so on. That is obviously still a very hot topic and very much debated. So this is how I designed the talk. I will give you a very brief update on the mutation landscape. I think throughout the talks at this meeting we have seen already quite some information. I will touch upon the more obvious actionable mutations for ALK and also MDM2. And then I'll take it one step further and start to think what we can do with the broader biology that we know about transcriptional deregulation in neuroblastoma, maybe some developmental aspects and so on and so forth, how we can exploit that towards drugging. And then, of course, there are some really interesting emerging targets that we heard at this meeting, H3, ATRX, and one of my old, of course, interests, the large copy number changes that I think that we can exploit. We have not really studied them at the functional level, and so I give you a bit of a flavor on that. And I'd obviously also very, very briefly will touch upon in vitro and in vivo models that are now expanding and will be extremely helpful to guide us through the choice of not only drugs, but in particular combination of drugs. So I will go very quickly here. I think most of you know this information. We have MYC amplification. We also have rare amplifications. You could call them smoking guns. So these are regions or genes that, well, most of them have turned out to be extremely important. Alclin 28B, MDM2, cyclin D1. We know overall, that was also nicely shown, that we have low incidence or frequency of mutations in most of the neuroblastomas. So let's say on the average less than 10, with ALK as being roughly the most frequent one, anywhere between 10 to 40% in the higher percentages in the high-risk cases. The work of Rohir's group on neuritogenesis-related genes, so I depicted that particular image here that you cannot read, but you know the paper, and many genes with very rare mutations, but all connected to differentiation. And then, of course, chromatin remodeling, which is an emerging topic, I think, in the field of neuroblastoma, and as already pointed out during the meeting, of course, very frequently implicated in other tumor entities with the ARID mutations, ATRX. And then I wrote here private mutations. I think that we now agree that we want to profile or sequence our primary cases, high-risk cases, see if we have actionable mutations, and some of them may be extremely rare. It was even pointed out here also for these GWAS hits, but they may have their relevance and they may be at some point used either in the relapse cases, if not in the primary cases in the further future, to drug them. So you probably also know these data. I think two very important papers that were published last year. I'm showing you circus plots, so they summarize all the data across the entire genome. So you see the chromosomes here listed, copy numbers here in green and red. The mutations, the genes are listed here on the outer side, and the structural rearrangements are listed on the inner side. So this is one primary case and the same patient at relapse, and obviously you see there's an increase in the number of genes that are mutated, an increase in the number of structural rearrangements, and so on and so forth. So this tells us that it's really important that we should try to biopsy. Maybe in the future we can do it by liquid biopsies to study what is happening at level of mutations, and also, of course, to use that information to further design therapies for in relapse cases. And as you probably also know, RAS-MAP kinase pathway is often targeted by these mutations that occur or are selected upon relapse. So that's coming to some of the issues that were addressed in Neil Rosen's talk. So we know ALK is a tyrosine kinase receptor. Yesterday there was this fantastic talk by Yael Mosse, sort of trying to understand what is happening if we're drugging this particular receptor. And I think, and that's what I wanted to address here in this slide, that we really need to understand exactly what is happening downstream in the downstream signaling. Why? Because we know that resistance to small molecules can occur either by mutations in the receptor or downstream mutations, for instance, in the RAS-MAP kinase pathway, and also what is called compensatory pathways, and that was alluded to in the talk of Yael and also of Neil Rosen. And this is really a big problem. You block this pathway, and then connecting pathways or pathways that take over the downstream signaling can be activated either by upregulation of ligands or whatever. So, I mean, that's sort of the biological system that is flexible, but that's a sort of biological basis that is co-opted by the cancer cells to resist to drugs, of course. And so with work that was published and then also presented here at a meeting, tried to dissect the downstream signaling of ALK to a further extent, showing feedback loops, which Neil Rosen again nicely pointing out that is really important, and a number of targets that might be druggable. So that's another point, that together with drugging the receptor, we might also look at downstream targets that could be interesting to exploit in drugging strategies. And we didn't present this here at this meeting, but we also found RET as a target of ALK signaling. So there's probably a very close and intimate relationship between ALK and RET signaling that we definitely have to further explore when we want to understand problems that may occur during resistance to ALK inhibitors. I put here image from a recent paper from the group of Rani George, where they created in vitro resistance to ALK inhibitors and then did further profiling. And there she found that, for instance, AXL, another receptor tyrosine kinase, was upregulated, just illustrating that these mechanisms readily occur and that we will have to take care of them in our therapy schemes. I don't think it was so prominent here at this meeting, but there was one particular abstract from our team work that was done, or I have to say by Jo van de Sompel's team, presented here by Alan van Gutten, and work that was done together with Jason Chauhet, where they showed that combination treatment of an MDM2 inhibitor, RG7388, together with a novel BCL2 inhibitor, when you combine them, you get very strong effects in reducing the tumor load and tumor growth in mouse models. So we know that MDM2 amplification occurs, and also that P16 locus encompassing ARF mutations or deletions quite frequently occur in neuroblastoma, and this is definitely, I think, also a target that we can further exploit for drugging. Well, these schemes are sort of simple stories telling you that in normal development, cell proliferation then stops during the process of differentiation, and there's a well-controlled transcriptional program. And that, of course, in cancer cells, this transcriptional program is perturbed. And that's something I think, obviously, we should further exploit also in our drugging efforts, not in the least our most obvious targets for MYC-N that is driving a lot of various cancer hallmarks through an entire transcriptional program. And also I think the epigenetic rewiring of pathways is something that we have to study further and exploit in drugging. To give you a bit of an overview, and I obviously don't have the time to go in all the options that we can exploit now for drugging, but I think part of it at least was summarized very nice in this clinical cancer research paper in 2013. And there you have ALK, of course, that we already discussed, and MYC-N, where we had this great talk by Martin Eilers in relation, amongst others, to Aurora A, but there are also other molecules that we can drug that are impacting on the stability of MYC-N, transcription factors like Lin28B that are involved in MYC-N levels and so on and so forth. So there's a whole range of drugs here that are listed here that we can start to use to target these mechanisms. And very briefly, we had an earlier paper by Kumps et al. Where we showed that in the MYC-N single copy high-risk tumors, there were actually focal gains or amplifications that were targeting genes that were downstream targets of MYC-N. And then one of them was, for instance, TERT, which then, as presented also here at the meeting, using further detailed studies with whole genome sequencing, was shown that was really frequently altered. And so I took this image from one of those from the paper of the Keulen group, where you saw the, let's say, division into three groups with the MYC-N amplified ones, the single copy ones with the TERT rearrangements, and then the ATRX, which has been, the deletions and the mutations have also been characterized and published. And now the challenge is whether we can also drug those entities with TERT activation or ATRX loss and alternative lengthening of telomere mechanism. And so there studies are ongoing to look for a synthetic lethality for ATRX deleted tumors, and obviously also antisense studies and so on and so forth are being performed in order to see whether we can target HTERT. And as a last part for this talk, I want to talk to you very briefly about the copy number alterations. You have seen probably this plot before. 17q has been for a long time our region of interest, very frequently gained. And as we pointed out also in the MYC-N-driven mouse models, we frequently see gain of a synthetic region for human chromosome 17, indicating that this copy number change is affecting the dosage of genes that are most probably very important to drive MYC-N-driven tumor formation. And we show that this meeting BRIP1, which is important to unwind stable quadruplex structures, as shown here. And just to show you that you can actually drug this mechanism, so there are ligands that stabilize these quadruplexes, and what you show here, I go very quickly here for reasons of time, but if you treat with this compound TIMP-P4, that you see there's also a drastic reduction in HTERT levels, so it might be a sort of indirect way to reduce the HTERT levels, and so we're further looking into also combination therapies with these ligands to see how they can be exploited for drugging the tumors. And you probably saw this plot, so we're also modeling that, so it supports that the copy number changes, they are affecting genes in their dosage that are important in tumor formation and that we can exploit for drugging. And then to wrap it up, what I think is really interesting is that apart from the wealth of biological information that is now coming towards us, we also have an increasing number of model systems that we are going to exploit to further study the biology and in particular to pinpoint druggable targets. So we have, of course, the classical cell lines. Now, of course, a lot of groups are focusing on the PDX. We have both mouse and fish-derived cell lines from the tumors that we're generating. And really interesting, and that's an emerging field, I think we have embryonal stem cell-derived progenitors that we will be able to use for modeling the disease. And then, of course, we have a whole range of new animal models coming up, both in mice and in zebrafish. So the take-home message, we talked about ALK. That's obviously one of our major targets for the moment. We have few mutations, some private mutations, but I think we really have to further focus on the signaling pathways and the regulatory networks. During our workshop, Stanley Key nicely pointed out that we will be able to use CRISPR technology as a really powerful technology to tackle these very complex gene regulatory networks. And as I said, I think we really, as a research community, we really have to focus more on these large copy number alterations and exploit them to identify novel targets for drugging. So we will probably first, and I think Lucas sorry, I don't get the name right, Moreno, is going to talk about that. How can we get the off-the-shelf drugs for drug repurposing? We have to understand these resistance mechanism. We have to combine that, of course, with other ongoing therapy, chemotherapy, the emerging and really important immunotherapy efforts. And then I just want to finish up. I don't have the time to go into detail, but personally I was really sort of triggered by this paper. And I would be really interesting to hear what the clinicians, the clinical oncologists think about it, but I find it a really sort of thought-provoking paper. Do we have to think the way we are treating the patients now in the context of the wealth of information which is coming towards us, the complexity of the regulatory mechanisms that are driving tumor formation, and probably also the variety of changes that are happening from one patient to another? Do we have to think of computational oncology, mathematical modeling of drugging, if we really want to go towards precision medicine? I'm a little bit tired because of the football game this morning, sorry for that. And I think also Neil Rosen pointed that out. I mean, we're moving to really complex schemes, and we don't have cohorts of hundreds of thousands of patients. And well, luckily I don't have to think about this problem, but I think it's something for the clinicians which is going to be an extremely big challenge. And I thought that was a nice slide to stop here, and I'm really ready to take some questions.
Well, thank you for that whirlwind overview.